Okay, now we have a look at the modes of transmission. So you've already had, so you've talked about pathogens. We have sort of been introduced to vector transmission when we've looked at malaria. We'll now have a look at the different modes of transmission, and there are essentially two main modes of transmission. There is a horizontal mode of transmission and a vertical mode of transmission. Horizontal is when it's direct control. I just like to think of it like horizontal as in like proper horizontal, and just helps me think of like direct contact. It can also include indirect contact. It includes vector transmission and contamination. So these four are part of the horizontal mode of transmission. Then we've got the vertical mode of transmission, and that includes transplacental transmission, vaginal birth, and also breastfeeding. So for example, with breastfeeding, if the mother has any pathogens like any pathogens, they can be transmitted to the child. Same thing, same thing with vaginal birth and also transplacental transmission. Okay, so now we look at two big experiments that really shaped the way in which the way that really changed our understanding of infectious diseases. Obviously, we now live, in a time where our understanding of infectious diseases and the technology that we are using to counter them has increased and evolved significantly in a really nice way. It wasn't the same as when the initial scientists who sort of set out on this path were doing their research. So the first scientist that we're looking at is Louis Pasteur, and Louis Pasteur is he is important because he disproved the theory of spontaneous generation. So people used to think that bacteria or pathogens just spontaneously infect things. He instead said that it's not spontaneous, it's not just happening randomly out of nowhere, there's a reason for it. And he proposed the germ theory of disease, which says that all things come from pre existing things. And to us now, that is like the most logical thing ever, right? But at the time, because they didn't have the scientific research to back that, they were just like, oh, it just happened. It can just happen. But he said, no, it is happening because it's coming from something that's pre-existing and that has contact with whatever it is that is being infected. And he shows that through his swan-necked flask experiment. So what he did is he took flasks with bent swan necks, and what happened was with the swan-necked glass, the particles in the air cannot travel through these necks without getting stuck. So if I've got something like, so firstly, let's start here. So I've got two different groups. I start off by applying heat to both groups. So by applying heat, initially what happens is I'm almost, I'm sterilizing it, I'm killing any bacteria in it by applying heat to it. I then let the flask sit for a while. But with one, I let it sit while it's closed, so while it is closed. But with this one, I open the flask, so I'm removing the neck and I'm letting it sit. When that happens, that means that suddenly all the microbes in the air have access to this solution right here. So as a result, all the microbes from the air are going to come in, and that is going to lead to bacteria being present in the flask. However, if I let the flask sit and I'm not removing it, I'm leaving it as it is, because of this passageway into the flask, you would see that there's no bacteria present because bacteria cannot, come in through here, travel upwards and go down here. It gets stuck here on the side. So as a result, there's no bacteria present. And yeah, so basically apply the heat, break the neck, observe the growth. Flask with broken neck means that there's microbe growth, and flask with without the broken neck means that there's no growth. And that basically tells us that it's not spontaneous generation. It's basically that the disease and the bacteria is coming from pre-existing microbes in the air that get access to these solutions right there. Okay. Next we look at Koch's postulates. So this is where he developed a procedure for isolating and identifying microbes which cause disease. So he wanted to sort of see what is, whether or not it is microbes that are causing disease. So what he did was, in all organisms with the disease, he says that, let's logically work through his steps. He said that in all animals that have a certain disease, there are going to be microbes present that are going to cause that disease. Those microbes can then be isolated and they can be grown in pure culture. So if I've got postulate 1 where the same microorganism, so I'm trying to prove the same microorganisms are present in every case of the disease. So the symptoms are the same. More than likely, there are going to be same microorganisms that are infecting these organisms. So here, my potential microorganism is the Anthrax bacilli. So what happens is I'm going to isolate that microorganism and grow it in a culture. So how do I isolate it? I isolate it from a dead from a tissue of a dead animal, and I make a pure culture. So I grow it in like an agar plate in my culture. Then what I do is I take the culture and I take the microorganisms from the pure culture that I've grown and I inoculate that into a healthy, susceptible animal, into another animal. Not the most ethical thing to do, but hey, that's what he did. So when we inoculate a healthy organism with a pure culture, it develops the same symptoms as the original organism. Because it's got those same symptoms, right? What that tells us is that ultimately it's that same microbe is causing that disease. However, what we do is we isolate and regrow that microorganism from that newly infected organism, and we do the process one more time. And so if same, then we realize that, hey, it's that microorganism that is causing the disease. So like I said, not the most ethical experiment, but that is how he was able to prove that microbes are causing disease. So every time you see similar symptoms, more than likely it's the same microbe. Okay, so we've already talked about this, but I'll just quickly go through this again because it is going to come up later. So direct contact, easiest way to catch the disease. You would know from COVID-19, from the pandemic. No shaking hands. We did elbows. Even fist bump was like not recommended. Always elbows, and so that is because there's that physical contact that's happening. Then we've got indirect contact, and that occurs when pathogens can live outside of host cells. So pathogens live in the environment after being spread. So something like influenza, where the pathogens are in the air, like after someone sneezes or coughs, the particles are in the air, and that can easily cause and someone can easily contract that. There's vector transmission, so insects like mosquitoes and ticks, which cause the Lyme disease, they can pass on the disease. And finally, contamination. So pathogens can be harbored in food and water, and there's abundance of nutrients because there's abundance of nutrients in food and water that encourages growth. With vector, we talked about transplacental, so the fetus can get a disease from the mother via the placenta, and examples of transplacental transmission include HIV and herpes. Vaginal birth, because there's microflora present in the mother's birthing canal, that can be passed on to the child during birth, and examples include sexually transmitted diseases. And finally, breastfeeding, so fluids and nutrients can potentially pass on infectious diseases through the milk to the child.